Hello, folks. Good whenever, because I can't predict when or where you're going to watch this video. But today we're going to be talking about plant anatomy, specifically leaves. So let's go ahead and jump over to our PowerPoint. So leaves are basically solar panels for the plant. They serve to provide surface area to capture the sun's energy and produce sugars by photosynthesis. So real quick, in a nutshell, what's going on here is the plant is taking CO2 from the air, water from the soil, and it is using the energy from the sunlight to assemble those molecules into more complex things like sugars. In doing so, it's storing that energy from the sun. So let's move on. So we need to be able to discuss what a plant looks like. That way we can talk about them intelligently with other people and start to classify them. So one of the first things that you'll notice when you're looking at a plant leaf is the venation on it. So does it have parallel or netted venation? So monocots will have parallel venation. So think grasses. And this might make you realize that grasses is a bit broader of a category than you might have formerly realized. So even corn, for instance, is a grass. Well, a monocot at least. And that's why when you look at bamboo, you can tell that it's a grass too, because if you look at its leaves, it's going to have parallel venation, meaning that the leaves running through those veins do not branch. If you have net-like venation, where it has a main vein with smaller veins branching off of it, then it is either palmately or pinnately net-veined. So palmate, like your hand, palmate like your hand, so think of maple leaf, or pinnate, where it's long, so pinnate like a right old-timey ink pen, because it's like a feather, meaning long and skinny, having all of these little branches off of the main stem, just like a feather, right? So your eudicots will all have this type of leaf. So when you look at a plant and it has net-like venation on it, you know that this is a flowering plant that produces seeds and possibly fruit. When you look at something with parallel venation, you know that you're looking at some sort of grass. Here are a couple of examples of this. This is our parallel venation. A good example that you might find in your backyard is an iris. Here's an example of our palmately net-like venation. And the easiest way to distinguish this from our pinnate is look at our pinnate at the base. We only see one large vein coming out of it all the way to the tip, whereas with our palmate one, we have 1, 2, 3, 4, 5. It doesn't have to be 5, it could be any number of main veins, but it's any number greater than 1, okay? And that would make it palmately net-veined. So these two are examples of eudicots. This is our example of a monocot. The next thing we're going to take note of when we're looking at a plant in the wild is the leaf Phylotaxi. In eudicots, so if you're looking at something and trying to decide what the Phylotaxi it is, this isn't very useful if you're looking at a monocot. So if you're trying to figure this out, make sure those leaves have some sort of net-like venation on them. And make sure you're looking at leaves and not leaflets, which we'll get into in just a little bit. So in eudicots, it can be either alternate, such as this, where you have one leaf on this side, then another leaf on the other side, one leaf on this side, another leaf on the other side, or it can be opposite, where you have two leaves found at each node. A node is a point where there is a leaf attached to the stem. So we see that there is a leaf attached to the stem at this point, and there's also another leaf on the other side of that point. So there are two leaves attached at the exact same point on the stem on opposite sides. So this is opposite leaf phyllotaxy or leaf arrangement. It can also be whorled, which is kind of a catch-all category for any time we have more than two leaves found at each node. It could be four like this, it could be a multitude, it could be three, although that's kind of unlikely. Point being, it's anytime you have more than two found at one node. Sound good? If you look closely in our drawings, we have some little nubbins at the bases of these stems, where the leaves meet the stems. These are our axillary buds. Those are how we determine that we are looking at a leaf and not a leaflet. So let's talk about leaf blade configuration. That's what we're getting into. So if we are looking at a leaf where you have your axillary bud, which will eventually turn into more leaves or flowers. So later in growth, if you see a leaf and then a bunch of leaves coming out of this little node in between the single leaf and the stem, you know that that was the original leaf, and that's what you'd use to identify the leaf phyllotaxy and the leaf blade configuration. Anyways, if your axillary bud is right there and you only have one single leaflet on there, then you have a simple leaf blade configuration, so a single blade. If you have an axillary bud and your stem goes out and then branches off into a couple of leaflets, we would call that compound. So it can be either palmately compound or pinnately compound. So this is just like the net-like venation. So if it branches out several leaflets from one stem, that is palmately compound, where it's at one point. And if it's all along one long stem where you have leaflets, that is pinnately compound. A good example of a pinnately compound plant that you might recognize is our state tree, the pecan tree. That is a pinnately compound leaf blade configuration. There are also bipinnately compound leaf blade configuration, where leaflets are divided into smaller leaflets and arise from various points on the petiole-like structures. So we have our one long stem, and stems branching off of them with pinnately compound leaves, it appears, but there aren't axillary buds right there. The axillary bud is way back here. So a good example of that would be our mimosa or silk tree, or for you folks out and further in west in Texas, a good example would be your mesquite trees. A good example of a simple leaf blade configuration for a lot of you folks would be an oak tree. Go out and find an oak, and if you'll look at the base of those leaves, there will typically be a little velvety nubbin, and that is your axillary bud. So here we have a sweet gum. This is our simple leaf blade configuration. It is also palmately net veined. Here is a pecan or hickory leaf. This is a pinnately compound leaf. This is a palmately compound leaf. And this is a bipinnately compound leaf. This is probably our silk tree or mimosa. This is potentially a buckeye. Just off the top of my head, I might be wrong on that one. So, Sometimes difficult to distinguish between leaves and leaflets. Remember to look for the lateral or axillary bud. So you see that, you'll find that right there. You'd find that at the base of this stem. So all tissue beyond a lateral bud constitutes one leaf. So plants obtain water and basic nutrients from the soil. So energy from the sunlight, and they use the light from the energy to make sugars via photosynthesis. So they're getting the CO2 from the air, they're pulling in from their stoma, water from the soil, they're using a few of their own enzymes, and they're using energy from the sunlight to assemble sugars, C6H12O6, and oxygen as a waste product. You might recognize that this is the opposite reaction of cellular respiration. So that's why plants give off oxygen and we breathe in oxygen and give off CO2. Likewise, that's why at nighttime, when plants are only conducting cellular respiration, breaking down these sugars, plants will consume oxygen instead of give it off. So let's talk about the anatomy of these leaves. So from the outside, they have an epidermis, which is just like in us, the outermost layer of cells. So epi on the very surface, dermis skin. So no chloroplast, which means they do not conduct photosynthesis. There may or may not be a cuticle, which is a waxy structure designed to hold in water and prevent water loss. If you want to see a real life example of this, a good example would be wax myrtle. It goes by a number of other common names, bayberry, heath, lots of things. Anyways, look it up, and if you want, you can probably find it somewhere on campus. Go out and rub it, and you can feel it's a little bit waxy, it smells fragrant. And if you were to want to make candles at home, which I guess is the sort of thing that we're all doing these days, you could take a bunch of those leaves, something with those waxy cuticles, not necessarily the wax myrtle, although it is commonly used for that, and you put them in the bottom of a big stock tank and boil it in water with a couple rocks to hold the plants down, and that waxy cuticle will actually liquefy, float to the surface, and then when you let it cool, you have a big old chunk of wax at the top that you can break off and then melt down and make into candles. FYI, if you ever see a bayberry candle, that's what they did. Apparently that used to be common in boutiques. I've never seen one though. So trichomes are these little hairs, plant hairs, that are designed to protect against herbivores, or in certain instances to prevent water loss. We're going to see an example of that in just a little bit. So let's look at the leaf epidermis. It's going to have stoma, which are pores through which the plants breathe. They're made by two guard cells. So the whole structure is the stoma. The two cells that make it up is the guard cells. If the plant is turgid, which means there is lots of water in the cells, which means it is sitting in a hypotonic solution. That's why plants always need lots of fresh water. The kidney bean shaped guard cells will swell and create the stoma, that opening. The plant loses water pressure, the guard cells deflate, and the stoma closes. The plant starts to wilt because it has lost that turgid pressure. And that helps to prevent water loss, but that's also why you see your plant wilt. That's why when you see your plant wilt, it's a sure sign that it's thirsty and you need to give it a little drink. Stoma allows the plant to breathe in CO2 needed for photosynthesis and allows it to release oxygen. Water can also escape through the stoma. This can be bad in certain circumstances, but this is also important in maintaining that your osmotic balances, because as water is constantly leaving those cells in the leaves, the concentration of solutes increases, making them hypertonic relative to the cells further down in the plant, which makes water want to go into the plant, go up the stem, carried out to these high concentrations of solutes to dilute them, and then providing them more water, which they're eventually going to turn into sugars or breathe out, repeating the cycle. So, few plants have found clever ways of opening their stoma at night, breathing in a bunch of CO2, storing it in a compound, and then shutting their stoma all the day long to help prevent water loss. These are known as things like C3 and C4 plants. You should look those up on your own time. I'm not prepared to talk about them right now. Very cool though. Those are typically desert plants, cacti and like. So, transpiration is the evaporation of water from the leaves of plants. Transpiration creates a constant flow of pressure up through the plant, pulling water and nutrients up through from the roots, just like I was just talking about. So this little opening that the guard cells form is the stoma. The two lips, the little pursed lips, making a little kissy face at you, those are the guard cells. These little puzzle piece looking guys, those are the epidermis. All right. So real quick, let's stop our share real quick. All right, let's go over to our real life example of this. So this is at 40x total magnification, and this is our leaf epidermis. This is probably our eudicot epidermis. So this is at low magnification. Let's increase that a little bit, get a better look at this. So this is very similar to the image that we just saw in the PowerPoint, but this is our real life example. The little puzzle piece shaped cells are the plant's epidermis. And you can see all these little pursed lips on the surface of it. And those are our stoma formed by guard cells. So let's pick a good guard cell and take a better look at them. I like that one right there. So here we are. Let's go ahead and snap a picture of that. That's a real good looking image. So here is our guard cells. There's one, there's the other. You can see the little nucleus in each one. There's a nucleus in one of these epidermis cells. The opening that they form is the stoma. And you will need to know this because I guarantee you it's going to make an appearance on the practical. Now real quick, let's go back to our video. Let me show you a much cooler image. So that was, I believe, our eudicot epidermis. It was all right. But let's check out this monocot epidermis because I think it is just handy. So this is at 100x magnification. It just looks good. Let me correct the hue a little bit. That way it looks more like what I'm seeing. All right, there we go. Here's another good example. Now, even though these are two very different plants, very different lineages, they're both going to have stoma, and they're both going to look pretty much identical. The epidermis cells are going to look a whole lot different. These look almost, yeah, these look almost like onion cells. Anyways, but these two guard cells forming the stoma look basically the same as these two. So you'll need to be able to identify your guard cells and your stoma. So stoma is the opening, guard cells are the ones on either side. So this is a nice turgid plant, got lots of water. So that stoma is open, it is breathing, letting out water, bringing in CO2, letting out oxygen, having a good old time. So let's get back to our PowerPoint. So mesophyll tissue. So if we take our plant leaf and we cut it in cross section, monocots are only going to have one type of mesophyll tissue, so middle tissue, meso, and that is spongy mesophyll. Like the name, it is spongy, full of little pores, not very well organized. Eudicots have two types of mesophyll tissue: palisade mesophyll, where it is very structured. They're rectangular columnar cells arranged in a layer, very organized, and that'll be on one side. And then they have spongy mesophyll on the other side. And so when you're looking at one of these leaves in cross section, you'll always be able to tell whether it's a monocot or a eudicot, by whether or not it has palisade mesophyll. Both have spongy, only one has palisade. Okay, so let's go ahead and look at our real life examples of that. Oops, got ahead of myself a little bit. So let's talk about the veins in these leaves. When you cut these leaves open, you're going to see vascular bundles in them. So in our monocot, they're going to be parallel. So you'll have a bunch of little vascular bundles side by side all throughout the leaf. So here's a bit of advice that's going to apply to all vascular bundles throughout the entire plant. So when you're looking at these vascular bundles, the largest pores are always going to be the xylem. Write that down somewhere. Beat this into your head. Okay, largest pores, always the xylem. And then we're going to have some smaller pores very nearby that are going to be the phloem. So here is our illustrations of them in these two different plants. Go ahead and take a look at what they look like in real life. So here we have a zeol leaf in cross section. Here's our vascular bundles. They look like little scary Halloween faces, right? So our two big eyes are our xylem, and then where the little mouth would be, the little screaming, horrific mouth, that's our phloem right there. You can see there's a little stoma. There's a little stoma with the guard cells on either side. See a little opening on the inside. Very neat. So let's look at our real life example of that. So that's it. There we go. That's better. Actually, there we go. So here is our monocot leaf in cross section. Let's get a better look at this, moving up to 100x total magnification. So here is our xylem. There is our phloem. All right. When we look at this, we scroll down along it. Oh, there's another vascular bundle. That one is in particularly good shape. So I'm going to go ahead and take a picture of that. Very cool. What a nice image. Xylem, phloem. And you see on the inside here, no palisade mesophyll. So let's go back to our image. See, we only have spongy mesophyll in here. Let's go to our eudicot. All right. So here would be our syringia. I unfortunately don't have that with me today, but I have a very similar specimen. You can see palisade mesophyll on the top, very organized, spongy mesophyll on the bottom, and then in the center, vascular bundles. Let's go back. So let's take a look at a real life example of this. So here we go. Here's our vascular bundles right here. And then let me scroll over a little bit. And you can see palisade mesophyll on the top, sponge mesophyll on the bottom. All right. So let's go back to our PowerPoint real quick. So your leaves are going to look a little bit different based on where the plant is growing. So in a plant, in a regular old normal running standard issue plant, okay, we're going to call it a mesophytic plant, and it's going to be outfitted for moderate climates. So doesn't have anything too fancy going on. Quite honestly, that plant that we were just looking at is a great example of one of our typical mesophytic plants. So let's take another look at this one as we go along that. See, just a normal standard issue eudicot, pretty much. Real quick, we have another example of a mesophytic plant here that we can take a look at real quick. So this is going to be the one that's going to look a little bit more like the one that you just saw in the PowerPoint. Let's turn off this vertical flip. There we go. See, just like the one you saw in the PowerPoint. Nothing too special about it. It's just a typical old eudicot. And you can tell it's a eudicot, not a monocot, because this palisade mesophyll on top, spongy mesophyll on bottom. So Next up, we have xerophytic plants. Let's take a look at one of those. They're outfitted for dry climates. They have cells closely packed together with stomata inside crypts or pits. And they also have long trichomes inside these pits that help prevent water loss by creating a little microenvironment where the relative humidity is higher than the outside environment. So let's take a look at that. All right, so here's a good chunk of our xerophytic plant to focus in on. So here you can see, I actually need to flip this upside down. So I have it upside down. Anyways, there on top we have our palisade mesophyll. Up here, you see it has a very thick epidermis, trying to prevent water loss, right? Because it's xerophytic, it's living in a very dry environment. And then what you're seeing over here is palisade mesophyll, and then a crypt, one of these little openings on the undersides. And that is full of trichomes, these long hairs. These ones that look like dots are ones that are coming towards you and got cleaved. Okay? And then somewhere in here, you would have your stomata. However, there aren't any just jumping out at me on that one particular one. Aha, there's one. See, right there, you have stomata with your two guard cells inside this little tiny crypt. Let's take a picture of that. That's a good one. Okie doke. So that is our xerophytic plant. Let's go back to our PowerPoint. So hydrophytic plants, they live in aquatic environments. They have few or no stomata. They also have a reduced amount of vascular tissue. So let's go ahead and take a look at that. Let's go back. There we go. We're going to have to zoom out for this one. Might even have to zoom out a little bit more. There's our vascular bundle right there in the center. Palisade mesophyll on top, spongy mesophyll on bottom. You might notice these giant, enormous openings down here in the spongy mesophyll. Now, why exactly would a plant do that, right? Well, if you live in water, well, see, plants they need a few things, right? Water, CO2, but if you live in the water's not really a problem. The gas is not really a problem either. What you do need, though, is sunlight, which is something that all plants are always fighting for. That's why vines are a thing. Anyways, if you live in the water, a good way to do it, instead of building up a strong stem and trying to prop yourself up, fill yourself full of air and float. So that's what a lot of plants do. So this spongy mesophyll would be full of air to help it float up. It has a little thin stalk reaching down into the muck to bring up nutrients. And that allows the plant to be very efficient and live in potentially deep or murky waters, things like that. So let's go back to our